Good morning, viewer. Welcome to our FTB English. Here's a breaking news. Much like humans, microbial organisms can be fickle in their productivity. So, one moment they are cranking out useful chemicals in vast fermentation tanks, metabolizing fit to make products from pharmaceuticals and supplements to biodegradable plastics or fuels, and the next they inexplicably go on strike. Engineers at Washington University in St. Louis have found the source of the fluctuating metabolic activity in microorganisms and developed tools to keep every microbial cell at peak productivity during biomanufacturing. The work, published in Nature Communications, tracks hundreds of Escherichia coli cells as they produce a yellow food pigment betaxanthin when growing, dividing, and carrying our normal metabolic activities. Like the behavior of a person, sometimes microbes are motivated to work hard, but they get tired much more quickly and easily, said Fuzhong Zhang, the Francis F. Hamann Professor in Energy, Environmental and Chemical Engineering and co-director of the McKelvey School of Engineering, Synthetic Biology, Manufacturing of Advanced Materials Research Center. Zhang is the corresponding author of the research along with PhD students Xinyu Mu and Alexander Stemets. Bioengineers and biologists have long observed large cell-to-cell variations in microbial metabolism, often called metabolic noise or more generally cellular noise. However, it has remained unclear what caused these differences and how frequently highly productive cells switch to low productivity states. And this lack of understanding has limited engineers' ability to develop effective strategies to enrich hard-working, high-producing cells for biomanufacturing. Answers lie in single cell fluctuating behavior, which is extremely challenging to study. So, researchers for the same must be able to measure a low abundant metabolite along with the enzyme that produces it inside a tiny single cell while that cell grows and divides. To address this challenge, the team built microfluidic devices and engineered Escherichia coli to produce a unique bright yellow metabolite beta-xanthin that can be easily distinguished from thousands of other cellular metabolites. These new advances allowed them to discover that beta-xanthin production fluctuates rapidly with cells switching from high productions to low production states within a few hours. There, approximately 50% of this beta-xanthin noise comes from fluctuation in the enzyme responsible for producing beta-xanthin, which arise from natural randomness, that is called stochasticity and gene expression. Fluctuation in cell growth rate account for less than 10% of the beta-xanthin variability. We have the exclusive report.